Hello students, I'm Dr. Jessica Han. In this section, I'm going to talk about protein. Proteins are the most diverse molecules that play important roles in our body. We have many kinds of proteins. Each kind of protein in a cell has a unique structure and a shape that allow it to play a specific function. We can obtain protein from our diet. Food such as fish, meat, beans, eggs are rich in protein. They are very good protein resources. Proteins are made by a chain of amino acids. Amino acid is the monomer of protein. When we link amino acid together, it will form a polymer. The covalent bond between the two amino acids are called peptide bond. As shown in this figure, each individual ball is amino acid. The whole chain of amino acid is a polypeptide. This polypeptide is the polymer of amino acid. This polypeptide can be twisted and folds, then lead to a unique three-dimensional shape. So the protein function is determined by the sequence of amino acids on this polypeptide chain. It also depends on the shape of the polypeptide can be folded into. So when this polypeptide chain be folded into three-dimensional structure, it will form a unique protein. For example, in this case, one polypeptide chain can form into a subunit of hemoglobin structure. When combine four of this subunit, it will form a functional complete hemoglobin. Not all the proteins need four subunits to become a functional protein. Some protein only need one polypeptide to be folded into a certain shape. Some protein need two subunits to form into a functional protein. Protein perform a huge variety of tasks. One of the tasks is structure. For example, keratin is an important protein, can be found in hair, skin, nails, and fur. Another function of protein is to transport. There are many proteins in our body function as a channel protein and a transport protein to transport molecule. The example of this is hemoglobin. Hemoglobin is found within the red blood cells. It carries oxygen and carbon dioxide and transport them through the whole body. One of the important functions of protein is to act as enzyme. Enzyme is a molecule can speed up the chemical reaction. For example, lactase. Lactase is an enzyme within our digestive system that can break down lactose in the milk. Protein can also function as defense because our antibodies are protein in the immune system that can fight with foreign invaders. Actin is an important component in the muscle, so protein can also facilitate with the muscle movement. The function of protein is determined by precise amino acid sequence and the shape of the polypeptide that can be folded into. For example, this is the normal hemoglobin, which requires four subunits. The normal red blood cell looks like a disc. However, when one of the genes is being mutated on the amino acid, then the red blood cells will look like moon-shaped. Then the sickle cell anemia will occur. Also, the shape of the protein will change once the mutation happens. We mentioned earlier, enzymes are important molecules of the protein. Enzyme can speed up chemical reaction without being changed by itself. We have many enzymes in our body, and those enzymes are important for chemical reactions in our metabolic system. The principle of how enzyme works is that it can lower the activation energy. Each enzyme has a specific shape, and each enzyme can recognize specific target molecule called substrate. For example, this is the lactase enzyme. This active site can only fit the substrate lactose, in which it has a complementary shape toward the enzyme. When the substrate binds to the enzyme will change shape. Then the chemical reaction occurs. The lactose is being broken down into glucose and the galactose in this case. Then the products are being released from the enzyme. Then enzyme return to its original shape. and is ready for the next substrate. The whole circle occurs again, and it illustrates that the enzyme can bind to its specific substrate to form into product. Enzyme can be inhibited. There are two different mechanisms that enzyme can be inhibited. One mechanism is called competitive inhibitors. The competitive inhibitors has a similar shape as the substrate, so that inhibitor can bind to the active site of the enzyme, but there's no product being formed. Another type of inhibition mechanism is called non-competitive inhibition. Non-competitive inhibition is that the inhibitors can bind to another site on the enzyme. Because enzyme is a protein, it has a three-dimensional structure. So the non-competitive inhibitors can find a spot to bind to the substrate. Even though the active site is still open, the binding of the inhibitor with the enzyme will change the shape of the enzyme. Therefore, the shape of active site will be changed. Then the substrate cannot come in and bind to the enzyme. Therefore, the products will not be formed. Therefore, like all proteins, enzyme function depends on its shape. Change in shape can prevent the shape can be changed by non-competitive inhibitor, and the shape can be changed by gene mutation, as we talked about in the previous slide. In summary, this chapter talks about the macromolecules. I have a summary table for you to use as a study guide. We have talked about 4 types of macromolecules: carbohydrate, lipid, protein, and the nucleic acid. Each type has their monomers. For example, carbohydrate, the monomer is called monosaccharide. When monomers link together, it forms into polymer. The polymer of carbohydrate will be called a polysaccharide. The examples of each type are listed here. The function of carbohydrate is immediate energy resource, and it functions as a structure and a support. There are some important notes I put it here for you to review the concepts related to the macromolecules. For example, what is a dehydration reaction? What is a hydrolysis reaction? What are the purposes of those reactions? I also list lipid, protein, and you can use this table to study the function and the structure of the macromolecules. In this chapter, we haven't talked about nucleic acid, which are genetic materials. We have 2 types of nucleic acid, DNA and RNA. We're going to learn their similarities and the differences in chapter 6. Hope this section will help you understand the structures and the function of macromolecules, especially protein and the enzymes.